Good morning, everyone. It is a beautiful, beautiful day down here along the beautiful, wonderful Schuylkill River once again. We're right off the Schuylkill River Trail, just outside of Roxborough, basically just north of Manayunk. We are now taking a follow-up sample. We did a sample a couple weeks ago during what's called base flow. That's regular river flowing. And now we're doing a storm flow sample, obviously because it's storming and raining. We are expecting, our hypothesis is that we will find more E. coli in the water at this time. Typically with runoff during a storm, there will be fertilizers from fields and residential areas and overly green lawns, people trying to make their lawn look like a golf course. We got pet waste in the streets and the curbs and the sidewalks. That's all running off into the river. Philadelphia has a combined sewer system we talked about last time, but we are still above where most of the outfalls are. So I don't expect it to be a huge jump, but I do expect to find some more. So we'll see what our results come up with. Our goals are to collect some samples in the rain, here we are, and to not fall in the river. Surprisingly warm.
Warm?
Yeah, that's wild. Maybe because it's colder outside in the field right now. So first we took the sample that we're going to be sending to Microbac, which is the lab. That's in the little plastic kind of like jar vial. We send that one off to a lab. It's up in Allentown. And then these are the plastic bags that we're taking back to our lab at the Schuylkill Center for analysis. Once again, we're going to incubate them overnight for 24 hours at 95 degrees and see what colonies might form this time around. So we're just kind of doing our own sample testing, but also getting professional lab work to cross-compare the results. Still looking very clear. But it's the stuff you can't see that we're interested in. So now we're going to be using distilled water as our control sample, just like last time, to hopefully, be able to really compare water from the river to pure distilled water. And as expected, that is clear as well. Anything else would not be a good sign. Hello, so we are back in the lab now, and we just plated the samples, put them in the incubator. So now we have our first storm sampling. We'll see how that goes. We'll see what changes based on the base flow results that we saw a couple weeks ago. And just wanted to show you this sample here. This was a sample I just plated. On the way in, unfortunately, I kind of tapped the side of the incubator and it lifted up the screen a little and just created these like air pockets here. So we're not going to be using this sample. Once again, my great lab assistant Eduardo Duenas reminded me that we should be using these as teachable moments. So all scientists and all people make mistakes, especially people who are very much learning, like myself. I'm very early on in this, and so I made a mistake. There's some air bubbles in there. We don't want that. We want it to be totally sealed so we can see the gas bubbles. So we're going to just go ahead and discard this sample, and our other 4 samples are in the incubator, and we will check back in 24 hours. How's it going, everyone? We are just finishing up looking at our storm flow samples what we took in the rain. And once again, as you could see in the slides, there are lots of little red dots which do signify different types of coliforms with the gas bubbles around them. But what we are searching for is specifically the blue dots, which signify E. coli. And our slides did not have any blue dots in them once again. So that's a good sign. It means the river is pretty healthy in this area as far as E. coli goes, even after the storm. We were sampling in the middle of a storm when it was raining, as you saw, but we're thinking also that next time we might want to wait till later in the storm or even after the storm, so that a lot more of the water can flow down from tributaries and other runoff from the area. We'll probably get a better sample then. The water was still clear when we took the sample, and typically, as people around the Schuylkill know, after the rain it definitely gets cloudier, darker, even brownish, kind of murky. So you know, good sampling. We're looking good. Our numbers were still lower than the other sites at Reading, Pottsville, and Bartram's. A lot of those sites are certainly closer to sewage outfalls, so of course that's going to greatly affect the E. coli numbers. But again, it's only been 2 samples, 1 base flow, 1 storm flow. We're going to continue to do this for 1 whole year, twice a month, 1 storm flow, 1 base flow sampling, and hopefully we'll have more information for you as time comes. But I really appreciate everyone from the community. Other folks reached out with questions, interest. It's really great to have lots of people involved in this project, so don't be afraid to reach out. And yeah, we'll try and keep you updated, and hopefully we'll see you out on the river next spring and summer, and hopefully we'll have some more updated data for you to tell you that around this particular area, the Schuylkill seems to be pretty clean and healthy. So thanks a lot, and we'll talk to you soon.